Hey, good morning. It's Mr. Peterson, just in case you were expecting someone else. I want to just take a couple of minutes and talk about the DNA structure or CER Google Slides assignment and make sure that everybody's kind of on the same page and that you're all aware of what you need to do for it. So there's a little bit of confusion that I've explained, so I thought I'd maybe throw together a screencast and see if that makes a difference to you. All right, so I'm on Power School, right? So I'm in the assignment block, and so there it is right there. Zooming, not zooming for some reason. Anyway, we've got these three documents here. And now we'll zoom. There we go. So there's the DNA structure, claim evidence, and reasoning directions. There's the DNA fact sheet, and there's the Google slide. So let's take a look at all three of those. So we'll start with the directions. So I open up the directions. So there's a little bit of introduction there, and some of the things we are already aware of. We've talked about genes, we've talked about chromosomes, and now we need to get down to the business of what is DNA. And so you're going to need, so if we look at the materials, there's a Google slide with the DNA model cutouts. Okay, make a copy of that. And you're also going to need the DNA fact sheet. All right, so that's still in Power School. The DNA fact sheet, read and use this data. All right, so you're going to need that information. Oops, that's something else. Sorry, my bad. All right, read through the directions here. It's important that you're working with the facts that are available. And so your model might look different than someone else. So the reason I'm asking you to do this assignment is the information that I'm going to give you on the DNA fact sheet. So all this information that's on the DNA fact sheet. This is what a couple of scientists, a guy by the name of James Watson, who was from the United States, he was a mathematician, and Francis Crick, who was a cat from Great Britain. He was a physicist, a molecular biologist kind of guy, and they were trying to figure out the structure of DNA. So they had a bunch of evidence. That's not true. They had a bunch of data that they were trying to turn into evidence. And so on the DNA fact sheet, this is the stuff they had available. This is the information that they had available. And so your job, so I'm on the Google slide now, is to take these various parts and arrange them into a model that's based on information on the DNA fact sheet. All right, so let's go right to the DNA fact sheet, which is right here, DNA fact sheet. All right, so number one, DNA is a long molecule, and it's made of repeating subunits. Okay, so it's a polymer. Poly means many. Mer, I don't know what that, but anyway, polymers are long molecules, and we've been seeing these all year long. Proteins, carbohydrates, whatever. DNA is composed of 6 subunits. So you'll notice that 4 of them are bases. So bases like in the opposite of acids. So guanine, thymine, cytosine, and adenine. There's another part of the molecule called a phosphate group. So it's a phosphorus atom with 4 oxygens surrounding it. And then a sugar molecule called a deoxyribose. It's 5 carbons, it's in a ring. That's about all you need to know, all right, about the sugar molecule. So you've got 4 bases, phosphate group, and a deoxyribose. That's what the subunits are made of. So let's look at that. So there's a nitrogenous base. C for cytosine, G for guanine, T for thymine, A for adenine, right? There's a deoxyribose molecule. So that's what those little cutouts are. And then these little goofy shaped things, those are phosphate groups. So in this model of cutouts, you're trying to figure out how this all goes together. Now to make a long molecule, because that's what the DNA fact sheet says, which is right here. Let's go back to that. Okay, number 3, DNA consists of 2 chains that are bonded together. So 2 chains that are bonded together. A subunit from one strand bonds to the subunit of another. So you've got 2 chains that are stuck together. Number 4, a sugar subunit can only bind with two other subunits, a base subunit and a phosphate group subunit. So a sugar, if we go to this, a sugar can only bind to a base and a phosphate. So sugars are never connected to each other. So there's some information. Sugars are not connected to one another. Number 4, I just read that. Number 5, a base subunit can only bind to two other subunits, a sugar and a base subunit. All right, so a base, this thing, cannot bind to another base. It can only bind to a sugar and a phosphate. Number 7, a phosphate group can only bind with a sugar subunit. So phosphates, this, can only attach to this. It never attaches to the nitrogenous base. So those are some hints on how to get things put together as far as the arrangement of the molecules. So it's a puzzle for sure. And here's what Watson and Crick were dealing with. Information like this. They actually used cardboard and actually tin cutouts. So instead of manipulating images on a Google screen, they actually used physical cutouts. And again, if we were in school, you'd be doing that with plastic cutouts. All right, number 7. Holy cow, what's going on here? Something's wrong with our image here. What's going on with our... what is that thing? Let's get rid of that. Whew. The diameter of DNA is the same along its entire length. So exactly 4 molecules or subunits wide. So it's got 4 subunits across. So there's a clue. Rosalind Franklin, this is Rosalind Franklin right here, made this discovery by using X-ray diffraction. And the way that works is you have X-rays. So this is some kind of tube, so like a light bulb that emits X-rays. So it's a different kind of light bulb. And they focused those X-rays by shooting them through a lead screen that had a hole. The focused beam of X-rays then traveled through the DNA sample, and just when light scatters through things that are not quite opaque, when they're kind of translucent, it scatters the light. And it wound up putting it on a piece of film, and this is what they developed. And so this X-ray diffraction, that's what that is. This shows these shadows represent places where there are molecules. And so you can see that there are places where there are molecules and then places where there aren't. And this is one of the key things that Watson and Crick used. All right, so we got to go back here. We know that the molecule across, or however you're going to arrange it, is 4 subunits across. So 4 of these things. So 1, 2, you're going to have to figure it out. You're going to have to, that's the clue. I mean, that's the fun of it. You could be wrong. Watson and Crick were wrong a lot. And that's okay. That's the way science works. All right. And then in 1950, a guy by the name of Erwin Chargaff he studied the proportion of bases in lots of different organisms. And he came up with this chart. So he noticed that the ratio of A's and T's and G's and C's, A to T and G to C, were always the same. So if you look at the human numbers, 30.9 and 29.4, that's pretty close to 30. And if we look at G to C, and you'll see it's 19.9 to 19.8. Those are almost exactly the same number. And again, some experimental error, right? And this is in 1950. 1950, right? So then he looked at chickens, grasshoppers, sea urchins, wheat, yeast, and a bacteria called E. coli. And he noticed that those ratios were almost the same. And so you're going to have to kind of draw your conclusion there and go back to this and see if that helps with that. All right, so that's kind of the nuts and bolts of how to make your model. And then you've got this business here. So your claim is going to be, I think the model looks like this. And then the reasoning statement, here's where you're going to go back and say, well, the reason the model has this shape is because when you put these things together, there are some rules. Oof, here are the rules right here. So that will help make your description in your reasoning statements. All right, so the reasoning statements really, you need to rely on the DNA fact sheet to help with that. Okay, so I hope that helps. I'll throw this up on PowerSchool, and hopefully that'll help you along with this. Shoot me an email if you're still stuck on that. But this is something you should be able to wrap up relatively soon. Okay, thanks. Have a good day.